basic genetics, and I've worked at the Children's Hospital of Philadelphia, actually for a long time, and I've been on their faculty since 1992. Part of my role now, today, is I'm head of our diagnostic division, our genomic diagnostic division. And one of the things that's been happening is that there have been so many changes in genomic diagnostics that, as you'll see, it's almost as though so many things have gotten better, but now it's harder to diagnose ring chromosome 20 because of some of the changes. And I'll tell you a little bit about why. So let me just start by saying the word genetics and what does that usually mean. So when you say that there's something is a genetic disorder, there are a lot of different ways for something to be a genetic disorder. One of them is for it to be chromosomal. Ring chromosome 20 is an abnormality of chromosome 20, so that's a chromosomal disorder. Chromosomal disorders can sometimes be inherited and sometimes not, and as you just heard from Dr. Canavini, often ring chromosome 20 is not inherited. So it's not something you've given to your children, it's something that happens. There are a lot of other types of genetic diseases. You can have a change in a single gene, which are often inherited, and diseases that you've all probably heard of, Duchenne muscular dystrophy or cystic fibrosis, some of the Tay-Sachs disease, some relatively common diseases. Sometimes it's not enough just to have a genetic change. You have a genetic change in combination with something, with lifestyle changes, obesity, or things that you eat, or something you're exposed to. There we have our regular chromosomes, and then a small bit called our mitochondria, which are only inherited through the mom, but we don't have to think about that here. Sometimes there are things like epigenetic factors. You're born with your chromosomes, and then they get changed because of your diet or exposures. Sometimes there are things caused by multiple different things that happen, and sometimes there is a whole group of things that are genetic, but we have susceptibilities to things like how we react to drugs. So genetics is kind of a whole wastebasket of things, and there are many, many. We're in the process now of trying to understand how all of these things impact human disease, but it can be complicated. So the Ring 20 group thinks of itself as having a rare disorder. You're not alone. There are over 7,000 suspected genetic conditions. And so in a place like CHOP, CHOP is the Children's Hospital of Philadelphia, so we're focused on pediatrics. And we, in our laboratory, make diagnoses for a lot of different diseases. And many of them are rare disorders. And, similar to some of the things that you go through, there are, groups who are trying to work to understand each of those. The estimate is that about 8% of live births are affected by some sort of a rare genetic disorder. So that's actually a pretty large number. In the United States, we estimate they affect about 25 million people and can be responsible for up to 20% of babies who die in the first year, so up to 25% of infant mortality. At CHOP, and like many pediatric hospitals, I don't think we would be any different. The estimate is that 35 to 50% of the hospital admissions are due to rare genetic disorders. And that makes sense because, as you know, once you have a child with problems, they tend to recur. So kids, and there are many different kinds of conditions where there are multiple surgeries and then you have to come back. That's not necessarily things that affect ring chromosome 20. Okay, so there are a lot of different ways of diagnosing a genetic disorder. The first test that was that we use for doing this is called chromosome analysis. And this came into practice in 1959 with the first demonstration that an abnormality of our chromosomes can cause a human disease. That was the recognition that Down syndrome has an extra chromosome. So what I have showing you here are, is there a pointer on here? No. Okay, I'm gonna step away. So what I'm showing you here, these are the human chromosomes lined up. So all of the DNA that's in every one of our cells is packaged into these chromosomes, into these 23 pairs of discrete, unique bundles of chromosome. If I looked at all of your chromosomes, my chromosomes, everybody's, pretty much we all have chromosomes that look like this. Girls have two X's, males have an X and a Y, but they're remarkably similar. at this resolution, so this is 3 times 10 to the 9th base pairs, 23,000 genes, all bundled up into these packages. It's really a remarkable thing, actually. I've been looking at them for a long time. I still think it's remarkable. So as chromosome physicians, which is, I guess, what cytogeneticists are, cytogenetics is cell genetics, so we look in the cell, but we're really looking at the chromosomes. We have techniques for getting them to have these banding patterns, but everyone sitting here, I'm sure, can appreciate some are big, some are small. They have different patterns of these bands, and people in our lab learn how to recognize those. So this is a normal female girl's karyotype, and we look to see, is something missing, is something extra. You can have everything go wrong you can imagine. You can have a piece missing, a piece extra, a piece moved, a piece pulled out and put in upside down. Chromosomes can pieces can come out and form unusual different structures. So there are a lot of different things that we can find. One of those things that we can find are ring chromosomes. So what is a ring chromosome? It is literally when the chromosome, which, as you saw here, they're all linear. They have a top and a bottom. But in a ring chromosome, it circularizes. So for some reason, you're actually getting the chromosome to just turn into a circle. How does that happen? I'll tell you what we know about it, but we can easily see this when we do a cytogenetic study, because it's quite obvious. And ring chromosomes can happen. They have been seen for every human chromosome. So ring chromosome 20 is not the only ring, but as you know, probably better than I do, it does form, there are certain things that happen, and so we can, it's become something that you can look at it and you guys have something in common because ring chromosomes give the same phenotype. But it can happen to every chromosome. This is a picture, this is called fluorescence in situ hybridization. And what we're showing you here in this picture is these dots, we make something that we call a probe. This is actually DNA. And we can actually take a piece of DNA from the ends of the chromosome and label it. We have laboratory ways of making it red or green. So we get a probe, so something we can go in and find, and I'll point to chromosome 20. We can get a probe for the bottom of chromosome 20 and a probe for the top of chromosome 20. And then we can ask, are those pieces there in the ring? So what you're looking at here is, here's a ring that has the top and the bottom, because you can see both the red and the green signal. So this ring, chromosome 20, just circularized, nothing was lost. Here's a ring that's missing the red part, which is, I think, the top, but is it the bottom? It's the bottom. That's missing the red part. Here's one that's missing the green part, and here's one that's missing both parts. So that's kind of lesson number one. All ring chromosome 20s are not the same. They can look different, and there are different ways we have of looking at them. So these are just some pictures of children with ring chromosomes, not all with ring chromosome 20, just to say that when you have all rings are not the same. And so there were a couple of papers. One of them was a paper from Laura's work when she and I were working on this several years ago. And these are actually from one of our collaborators in Brazil. All of these patients have ring chromosomes. They have different problems. Many are much worse than you see with ring chromosome 20. And this is a function of which chromosome is involved and how much of the chromosome is missing when that ring gets formed. So that's kind of lesson number one, or all rings are not the same, or you can have rings for every chromosome. All rings are not the same. And what you see clinically depends on which genes are involved and which chromosomes are involved. Okay, so as Dr. Canavini said, ring chromosome 20 was first described in 1972. From 1959 to the 1980s, everybody was using chromosomes to try to understand different syndromes that their patients presented with. So there was an explosion of people doing chromosomes, and here was the first finding. A 21-year-old woman with a ring 20 chromosome is described, and in this case, the patient had behavior problems, epilepsy, low grade, we now say intellectual disability. are the same as in the 3 cases described earlier. So this now forms a syndrome. A syndrome is when you have a recognizable constellation of features. And here you can see the ring chromosome 20. This is a picture that was from our lab. So we already heard what is involved in the syndrome: pharmacoresistant epilepsy, cognitive and behavioral problems, intellect often normal prior to seizures. So we'll come back to the fact that early diagnosis would be really important. Mild to moderate intellectual disability. Non-dysmorphic means that you don't really recognize different features of the face. Dysmorphia is when your facial features have some deviate from what we call normal. Looking around the room, everybody knows that we're all normal, we're all different, but clinical geneticists have come up with ways of looking at patients and saying, is this a normal face? Are the eyes too wide, or is the face too long? So for a lot of syndromes, if I showed you pictures, after sitting here for an hour, you'd be able to recognize them. That's not true with ring chromosome 20. And generally, there are no congenital malformations. A lot of genetic syndromes, you're born with a heart defect or a bowel defect or abnormal limbs. So we don't see any of those in ring chromosome 20. Okay, so the presenting feature is epilepsy. And how do we diagnose epilepsy today? So we actually have Ingo Helbig and Ahmed Abu Tayoun. Ahmed is in our division, and they've been working on epilepsy diagnosis. And they have put together a number of reviews on this now. There are hundreds of genes for epilepsy. So when a child is diagnosed with, when a child starting to have seizures and you want to do diagnosis, they look at hundreds of genes. And this was a review that they put together showing you since 1995 all of the different genes that have been identified for epilepsy diagnosis. This work is done since the Human Genome Project started, so that we were able to find all of these new and different types of genes that cause epilepsy. None of these, of course, are related to ring chromosome 20, but there are hundreds of genes that cause epilepsy. So if a patient appears who has epilepsy, 15 to 20% of patients are going to have mutations in some of these genes that have been established. We are still finding new genes for epilepsy every day. So another 5 to 10% are going to have these new genes. There are no genes that a lot of patients have. So epilepsy is a really hard thing to diagnose because there are so many different reasons that you can have epilepsy, and the types of seizures and some of the accompanying features are very different from one to the next. And as Ingo points out, Ingo is a neurologist who does a lot of epilepsy research. When he sees a patient with seizures, he usually cannot guess what their cause is going to be. So the neurologists tell us that it's very hard to diagnose epilepsy. And I just read on the plane ride here, and I didn't have time to put it into any of my slides, I read a new paper that just came out talking about 670 patients who were newly diagnosed with epilepsy. They did genetic testing on a little over 300 of them. These were all patients who had early onset epilepsy before the age of 3. They found, and they did all of these different tests. 57 of them had chromosome analysis. There were no cases of ring chromosome 20 in that test. Diagnosis before the age of 3. So this is a really complicated field, and part of us has always been very frustrated that ring chromosome 20 gets underdiagnosed, but it's not really surprising given the complexity. Okay, so back to ring chromosomes. We know that ring chromosome 20 causes epilepsy. What's actually going on? There are a few of these ring chromosomes, I told you we've seen them for every chromosome, that have had seizures involved with them. Ring chromosome 20 isn't the only one, but if you look at, this is from a paper in 2006, if you look at, these are all the different ring chromosomes, ring 20, ring 14, ring 17, ring 6, and I'm looking, ring 4 isn't even on here, we just saw one of those. The most commonly, ring chromosome 20 and ring chromosome 14 cause seizures, along with ring chromosome 17. we've studied both of these different disorders. We've seen very few ring chromosome 17s. I think only one, two. So pretty rare. Our lab does a lot of cytogenetic studies every year, in excess of 2,000, and I've been there since the early 1990s. So how do you diagnose ring chromosome 20? I'm going to show you why, but chromosome analysis is required, but chromosome analysis is not necessarily part of the standard workup. Now that may be different for people who are very, very familiar with the syndrome, but today, when the physicians, and we went to talk to our neurologist several years ago with Dr. Hossein, who couldn't come here today, and we tried to explain why it's so important to do chromosome analysis. But the data that I just showed you, the complexity of epilepsy, how many different genes there are, I think has put people off, and the neurologists don't, and ring chromosome 20 is rare, it's not the most common cause, so it's hard to get people to order chromosomes. It's another test, none of these tests are cheap. So we recommend that you order chromosome analysis separately when you think that the patient could have ring chromosome 20. So what's happening when that ring chromosome forms? And this is just a diagram, a cartoon really, to show you what you have this chromosome and it's linear and it's going to form a ring. How does that happen, and what happens to that chromosome when you form that ring? So there are several different possibilities. Possibility number one, the whole thing is there. For some reason, the ends just get sticky. All material is intact, and the chromosome circularizes, right? Nothing lost, nothing gained. You just have, for some reason, the ends sticking together. That's possibility number one, and that, in fact, does happen. Possibility number two is that you have these, and you have this green part here, which is the very end, and these just stick together, which is shown here. Possibility number two is that there's this subtelomeric region just under the telomeres. You lose this part, and you end up fusing into that region. So a little bit comes off at the end. And possibility number three, well, I guess here you're just fusing at the telomeres. Here you're just losing one or the other ends. Here you're losing both ends. And here you're going deep into the chromosome. So you're losing nothing at the end, something from one side, something from the other side, or something from both sides. And as I pointed out before with that picture, we have seen all of these different things. So can we diagnose these different situations is question number one. So in 2005, about 2004, 2005, the world of human genetics was advancing, and a new technique came out. And I'm showing you some data from our lab for chromosomal microarrays. How many people have heard the term chromosomal microarrays? No. How many has anybody's child had a chromosomal microarray for diagnosis? Yes, you probably have. So cytogenetics is old-fashioned, looking at the chromosomes in that big way. What we're looking at here was the big change, and actually the paper that I just read this morning on epilepsy diagnosis, they went through all of the different tests that their 327 patients had. Chromosomal microarray was the most common. So this is today one of the most commonly used tests if you think a patient might have chromosome abnormalities. And what this is showing you is this is a picture. This is a diagram of chromosome 1, chromosome 4, chromosome 13. Every one of these dots is a particular position, and this diagram is telling you, do you have 2 copies of that position on chromosome 1 as you're supposed to have? And without going into all of the details, here's a normal chromosome 1. You can see what an abnormality looks like. Here's a chromosome 4. This is all, it's a little messier, but see right here? There's too little of this part of chromosome 4. See this one? There's too much. This shows you that there's too much, and you don't even have to really worry about it, but you can just look at it. If I just kept showing these to you, this is normal. that's missing, this is extra, and here's something here that's a piece that's extra. So this technique changed our lives in our lab. We went from doing 2,000 chromosome diagnoses a year to doing 2,000 SNP arrays, chromosomal microarray analysis per year, because this test is so much more sensitive, except for ring chromosome 20. And why is that? So in the middle of 2006, I think it was, Laura and I started working on ring chromosome 20, and the first question we wanted to ask is, what's wrong with those rings? Show us what are those rings made up of? And we started doing a few types of studies. We did chromosome analysis to identify the rings, and we did FISH to see what was missing, and we did chromosomal microarray to see how much was missing when something was missing. So here's an example of one chromosome 20, and we made an interesting observation that we still are kind of surprised by. We found two different types of rings. You all heard the word mosaic before, right? So the first group of rings had a ring chromosome in every single cell. You looked at the karyotype, there were no normal cells. There was no two normal 20s. Every cell had a ring chromosome 20. When we did FISH, all of those rings were missing something. They were different. Here's one missing both ends. Here's one missing one end. And here you can see it on the array. Here's one something has a deletion at both ends, right? Down here, this is chromosome 20. From the top of it all the way down to the bottom, missing something here, missing something here. That's a non-mosaic ring, every single one of them. So what does it mean that it's non-mosaic? That means that when your child was being formed, that first egg meets sperm, and you have that first zygote, we call it, right, that fusion, right there, that ring chromosome 20 already existed in that cell, and then throughout, we go from one cell to 10 to the 14th cells. Every single cell has the ring 20. So that's the first example, but those ring 20s are different. Sometimes missing more, sometimes missing less, sometimes missing one side, sometimes missing both sides. And here, this is a paper, just a figure from the paper that we wrote in 2010, and again, just showing you this again. Sometimes, and no deletion, one side deleted, both sides deleted, the other side deleted. Then we looked at the patients who were mosaics. So what does that mean? That means that we look at 30 cells, and 2 have the ring, the rest don't, or 10 have the ring, and the rest don't. But the cells that don't have the ring are totally normal. So here's a mosaic patient, and this is so in this one young woman, there were 2 chromosome 20s. In some of her cells, they're both totally normal, and in some of her cells, there's a ring and a normal chromosome, right? So it turns out that 2/3 of the patients are mosaics. In every single mosaic that we looked at, and this is just an example, if you could see, so I'm showing you, this is a calico cat, who's one of the best examples of a mosaic. Why? Because you can see it in the cat's skin. Most of us, we could be mosaic for many things, but we don't see it. If you were mosaic for a pigment gene, you could see a blotchy pattern, and that's what you see with the cat. That's why we use it as an example of a mosaic, just because the changes you can see on the cat's skin, and you can see these different kinds of fur. So that's what a mosaic is, and when you're a mosaic, you have different patterns of cells all throughout your body. So what we observed is that all of the mosaics, there are no deletions. Those rings, as far as we can tell, have all bits of the genetic information that are intact. So this was really interesting to us. One, because every non-mosaic, every patient, if it's in every cell, is missing something, and every mosaic has nothing missing. So that surprised us and gives us a clue about how these happen, but we haven't been able to use that yet to understand why those chromosome 20s, why do rings form in those cases. But nevertheless, it's kind of interesting. It's also a clue about what's going wrong, because it's not just what you're missing. We know from our cytogenetic studies that deletions usually cause trouble. Or deletions, there are many, many genes that you can't have in only one copy. That having one is not good enough. And so in many different cases, you get disease because of that. But in this case, we have a phenotype. The kids have seizures, but it's not because something's missing. So there has to be a different explanation for that. So we have been trying to understand what that could be, and our current work is focused on trying to understand that. And you'll hear a little bit more about our current work this afternoon from Laura. So there are a few things we do know about rings. One, rings are really unstable. They just don't do well in the cell. So one possibility, and this is something that we've noticed, is that the ring gets lost. All of a sudden you're looking at cells, and for those of us who sit at the microscope, and you've got some cells with no ring in it, like where'd the ring go? It's missing. So the cell's not going to like that. That could cause some trouble right there. Sometimes the ring duplicates itself. And this is because a chromosome is supposed to make a perfect copy in order to duplicate itself. But when you've got a ring, it's just physically, I think it gets hung up, the DNA, and I'll show you some lovely pictures, but showing you what happens when this ring tries to duplicate itself. And sometimes it opens. And this is a ring chromosome that's been, something comes along and breaks it, and it opens. And now you have a funny looking chromosome 20, but it actually used to be a ring, but it's opened up, and we've seen that once. It's hard to recognize in the cell. So what's causing problems in these kids with perfect rings? Is it loss of sequences at those fusion points? We don't think so. This was the list we made before we had any data. We said, okay, maybe you're missing things, and those things are really important. But our data kind of showed us that's probably not the answer, because not everyone's missing something. Maybe it's just these rings are having so many problems, and it's just the mechanics. You know, the cell can't quite divide, or the cells with the rings are dividing at a slower rate or cause problems. Maybe it's the cells that have lost a ring and they only have one chromosome 20. Maybe that's causing problems. Maybe there's something about being a ring that causes, the job of chromosomes is you have all these genes on you, and your job is to make copies of those genes in order to turn them into proteins that do the work of our bodies. So it's really the proteins that make up all of our organs and do all of the work of our body, but the information for it is coded in our genes, and you make RNA, and then you go ahead and make those proteins. Maybe when you're a ring, the signals to make the RNA get faulty. This is a term, uniparental disomy. This is a term that means sometimes instead of having one chromosome from mom and one chromosome from dad, you end up with two from the same parent, which is odd and sounds odd, especially to people who aren't familiar with the term. But in fact, in some of our cell culture models, we know this is exactly what happens. And we'll go into that a little bit more later. But this is another possible mechanism. So this is kind of where we started, and we wanted to try to figure out which of these things was causing trouble. This is the diagram I was showing you. This is from a really nice paper from Roberta Gilherm, who works with Isabel Melorano. And they just were doing some studies of rings, and you can just see what happens. So here's a chromosome 10. This is a ring 10, but there are two copies in there. It's, you may not be able to appreciate. Sometimes there's just an extra copy of a ring. Here's one with a double ring, and then looks like even a triple ring. And here's one where the ring chromosome has broken open and is now abnormal. So all of these different things can happen. And this is the example of uniparental disomy. This is from a paper that that was published a few years ago by another group working on chromosome 17. And we've seen the same thing. We have a lot of data on this. Laura's going to show you some of it later for ring chromosome 20. You start out with a ring, and then as you try to make a cell culture model, the ring gets lost and you duplicate the normal chromosome. So they thought this could be an exciting way to think about correcting problems in the cell. We think it's a little more complicated than that, but we want to be able to set up a cell culture model to study this, and it's been very complicated. So conclusions to date, and again, this is the history and where we've been, and this afternoon you'll hear about our current work. Ring chromosome 20 is associated with consistent features, as you all know. You need chromosome analysis to diagnose this abnormality. The newer technique of chromosomal microarrays will not diagnose the 2/3 of patients with a mosaic ring. A third of patients have the ring in every cell. These will be diagnosed by array analysis, but 2/3 have mosaic rings with no deletions. So our goals have been, first of all, to focus on developing a cellular model, which has been very tricky, and you'll hear why. We want to understand how the ring causes the clinical problems that it causes, and the goal here has been to there are new techniques for making, they call them organoids, brain organoids, so you try to turn cells into neurons that form some sort of a brain-like structure, and we've been working on that with a collaborator of ours. And we're hoping that once we have a cellular model, you're able to test more drugs, because it's hard to do that in people, but if you have a cell culture model. So I hope I didn't go too far over, but in conclusion, just to say thank you. We, of course, have a team of very talented people who we've worked with through the years. Laura, Debbie McEldrew does all of our cell culture. Jay Mills developed all of the IPS cells. Stuart Anderson is our current he's not a neurologist, he's a neuropsychiatrist, but he's a neurobiologist who's been helping us with all of the models. And Melissa Gilbert, Ronke's work you're going to hear about a little bit later. So thank you very much.